My name is Pritibha Dwivedi and I am working as Assistant Professor in Biyani group of colleges. I am here to present a video lecture on behalf of gurukpo.com. Today's topic is transport across plasma membrane. The main focus of this lecture would be on the passive diffusion type of the transport. As we all know that plasma membrane is made of the phospholipid bilayer. This phospholipid bilayer has two domains, hydrophilic head domain and the hydrophobic tail domain. The hydrophilic head domain is made of alcohol, phosphate and the glycerol group. These groups made this domain as a hydrophilic in nature, and the fatty acid tail domain is hydrophobic in nature. To maintain the internal environment and internal composition of a cell, it is necessary for a cell membrane to be selectively permeable. It means it allows diffusion of some molecules across the plasma membrane. The plasma membrane transportation may be the transportation of molecules across plasma membrane is mainly of two types, the passive diffusion and active diffusion. Passive diffusion is always along the concentration gradient. It means जिस तरफ concentration ज़्यादा होगी उस तरफ से molecules कम concentration की ओर move करते हैं. Molecules कम concentration की ओर move करते हैं. Transportation of molecules across the plasma membrane is divided into two types, active diffusion and passive diffusion. Passive diffusion always occurs along the concentration gradient. It means the molecules will move towards the lower concentration across the plasma membrane. Passive diffusion constitute of two types. It depend on the concentration gradient and the electric potential across the plasma. The transportation of molecules across plasma membrane is mainly of two types. These are, first is active diffusion and second is passive diffusion. Passive diffusion does not require any metabolic energy to facilitate the transportation of molecules across the plasma membrane. This passive diffusion is mainly of two types, simple diffusion and facilitated diffusion. The active diffusion is also further divided into two types, primary transport and secondary transport. Active diffusion requires metabolic energy to facilitate the transportation of molecules across the plasma membrane, unlike passive diffusion which do not require metabolic energy. Passive diffusion also always occur along the concentration gradient. It means the molecules will move towards the lower concentration of their across the plasma membrane. The passive diffusion depend on two things. First is the concentration gradient and second is the electric potential across the plasma membrane. The electric potential always occur in the case of charged molecules. The passive diffusion is always occur along the concentration gradient. It may be of simple diffusion. It means the molecules simply dissolve in the lipid bilayer and move across the plasma membrane. And it can be facilitated diffusion. It means the carrier proteins or channel proteins facilitate the diffusion of molecules across the plasma membrane. We all know that plasma membrane had the hydrophilic head domain and the hydrophobic tail domain. So the requirement is when the synthetic lipid bilayer is checked for the diffusion of molecules across the plasma membrane, it was observed that some uncharged and small charged molecules can simply diffuse across the plasma membrane through the simple diffusion, while some large charged molecules do not simply diffuse across the synthetic bilayer membrane. Thus, it is observed that some proteins and some other mechanism are involved in the transportation of molecules across the plasma membrane. As this figure show, the synthetic lipid bilayer only permit the diffusion of hydrophobic molecules like oxygen, CO2, nitrogen and benzene. It also permit the passage of small uncharged molecules like H2O, urea, glycerol, etc. But large uncharged molecules, for example glucose and sucrose, also moves across the synthetic bilayer of lipid membrane, but the movement is very slow across the membrane, and the synthetic lipid bilayer do not allow the passage of ions like sodium, potassium and chlorine ions. But in the natural occurring cell membrane, it is observed that ions also move across the plasma membrane. Thus it was concluded that some other proteins are involved in the transportation of molecules across the plasma membrane. Now focusing on the passive transport method of the plasma membrane transportation, the passive transport always occurs along the concentration gradient and it does not use any metabolic energy for the transportation of molecules across the plasma membrane. The passive transportation is always influenced by the two factors. First is concentration gradient across the plasma membrane and the other is membrane potential. The membrane potential is the electric potential generated by the relative concentration gradient of the charged molecules inside and outside of the plasma membrane. These both gradient are combinedly called as electrochemical gradient. The passive transport further divided into simple diffusion and facilitated diffusion. Simple diffusion is in this method the molecules simply dissolve in the lipid bilayer and moves across the plasma membrane. This simple diffusion depends on the relative concentration of molecules inside and outside of the plasma membrane. It does not use any molecular protein or any other system for facilitating the molecules to transport across the plasma membrane. But facilitated diffusion, it requires some proteins to facilitate the diffusion of molecules across the plasma membrane. This facilitated protein may be of two types, carrier proteins and channel proteins. Now first coming on the mechanism of simple diffusion method. Simple diffusion is a non-selective method of transportation across plasma membrane. It means any molecule which can dissolve in the phospholipid bilayer can easily move across the plasma membrane. The relative diffusion rate of any molecule depend on the concentration gradient across the plasma membrane and the hydrophobicity and size of that molecules. The hydrophobicity is measured by the partition coefficient. It means the equilibrium constant for partition of any molecule between water and oil. It means the molecule how parted itself in the water and oil. The partition coefficient increases, the lipid solubility of molecule increase along with. The rate of the diffusion can be measured by the Fick's law of diffusion. The scientist Fick give the diffusion rate by the equation J is equal to minus D delta C upon delta X. Here J represents the flux per unit area. Negative sign represents that molecule will diffuse along the lower concentration and toward the lower concentration of its, and the delta C is the difference in concentration across the plasma membrane. Delta X is the membrane thickness and capital D is for the diffusion coefficient which is measured in centimeter square per second. The simple diffusion is a non-selective method of diffusion along the plasma membrane, which which means the any molecule which can dissolve in phospholipid bilayer can easily move across the plasma membrane. So just like uncharged molecules, for example some gases, oxygen, nitrogen and CO2 and benzene, and uncharged molecules like glucose and sucrose, urea and water, etc., can move easily by simple diffusion method. Now coming on the facilitated diffusion of transport. Unlike simple diffusion, the rate of transport is greater than in facilitated diffusion than simple diffusion. This also occurs along the concentration gradient across the plasma membrane. The facilitated diffusion allows polar and charged molecules to move across the plasma membrane. It means unlike simple diffusion where only uncharged molecules can move across the plasma membrane, in facilitated diffusion the charged and polar molecules can also move across the plasma membrane. The facilitated diffusion can be facilitated by two types of proteins. First is carrier proteins and second is channel proteins. The carrier proteins, also known as transporters, involves in non-covalent binding of the molecules, and on the binding to the molecules they undergo in a conformational change, and after going across the plasma membrane it releases the molecules outside the plasma membrane. The rate of transport in carrier protein mediated transportation can be given by the formula equation V is equal to Vmax upon 1 plus Km upon C. Vmax here is the maximal rate of the diffusion. The Km is the concentration of substrate where the rate of diffusion is half of the maximal transportation rate, and C is the concentration of substrate. This equation is equal to the Michaelis-Menten equation in the enzymology, where we examine the rate of equation which is catalyzed by enzyme. Here this equation gives the rate of diffusion across the plasma membrane. The channel proteins mediated transportation may be of two types. First is voltage gated proteins and second is ligand gated channel proteins. Voltage gated channel proteins open the channel in response to change in the voltage or electrical potential across the plasma membrane, while the ligand gated channels open the channels inside the plasma membrane in response to the binding of any ligand. These channel proteins form open ports through the plasma membrane along which any molecule can go through across the plasma membrane. The rate of diffusion in channel protein mediated transportation is more higher than the carrier protein mediated transportation. The inorganic ions transporters involved, the channel proteins involved in inorganic ion transportations are known as ion channel proteins, while the aquaporins are those channel proteins which are involved in the facilitated diffusion of water molecules across the plasma membrane. Thank you for watching this video. Please share your comments and likes on this channel. Thank you.